I'd like to begin by thanking the organizers of this year's conference for the invitation to speak to you today. The title of my talk is Computational Tools for the Identification of Unknowns. And of course, when we talk about unknowns, we often think about a quote that appeared about 17 or 18 years ago by then US Secretary of Defense Donald Rumsfeld about what we don't know. And his quote is famous for a few things, but in particular, he mentions this: there are known unknowns, that is to say, we know there are some things we do not know, but there are also unknown unknowns, the ones we don't know that we don't know. This is a very telling quote because it's actually been a term or set of terms that we use frequently now in metabolomics to talk about the unknowns that we often have to deal with. This is a graph that's illustrating, if you want, the unknown unknowns, or we call it the dark matter of the metabolome, and the known unknowns. So at the bottom corner, left corner, is an illustration of our sensitivity and the typical number of metabolites that we can detect using different technologies in metabolomics: NMR, GCMS, and then LCMS. And as you can see in the lower left corner, NMR is perhaps the least sensitive technique, typically with a lower limit of detection of 10, maybe 15 micromolar, and a number of compounds that it can detect around 50 to 100. Many of the compounds that we see in NMR are actually knowable or known. They just, at the time that you collect your spectra, are going to take a little bit of effort to figure out what they are. So these are the known unknowns. That is, we have information about them, there are references that we can identify those spectra with. And in most cases, 95, even 100% of the peaks that are seen in an NMR spectrum can be assigned. With GCMS, you're in a similar realm. Many of the peaks can be identified, perhaps not 100%, maybe 55 to 65%. The sensitivity for GCMS is a little better than NMR. It's sometimes submicromolar. The number of compounds that can be identified by GCMS is often in the high hundreds, sometimes up to 200. Of course, most people prefer the ultrasensitive techniques that you'll see in liquid chromatography mass spec or direct injection mass spec. And in these cases, often 10,000 or more features are identifiable. These correspond to peaks in the chromatogram as well as mass spec peaks that are detected in the total ion chromatogram. With mass spec, we're often able to get down to the low nanomolar, even picomolar sensitivities. And as I say, it's often to be possible to identify several thousand features, but most of these features are not identifiable. And most of those features fall into a class that we call the unknown unknowns, or the dark matter of the metabolome. Now this gets into another realm of metabolomics, which is the two types of metabolomics that we typically use in the field. One is called targeted metabolomics, where there's a formal identification, quantification of a set number, pre-identified collection of metabolites, usually on the order of 100, maybe 200, 300 compounds that are targeted in some of the larger labs or with some of the kits that are now available. The majority of people in metabolomics still pursue untargeted metabolomics, and this is a process where the intent is to cover as many possible identifiable or unidentifiable features, to use high-resolution mass spec, to use a variety of tools to identify as many peaks as possible, to use statistical methods to winnow that down, but then again to be left with the task of identifying literally thousands of peaks. So in untargeted compound identification, it's typically described with this sort of simple picture. A total ion chromatogram might be collected. Under any given peak, there could be a few to even dozens of overlapping compounds with their own unique MS-MS spectra, or if you're dealing with GCMS, it would be their EIMS spectra. Each of those unique and hopefully pure spectra can then be mapped to a database of MS-MS spectra. And by comparing the similarity of the MS-MS or EIMS spectra to this spectral database, it's possible to identify the specific compounds in the untargeted technique. Of course, all the spectra are tied to specific structures, and from there we're able to hopefully identify dozens, hundreds, even thousands of compounds. Now, the process for metabolite identification is often graded. Metabolomic Standards Initiative, or MSI, came out in 2007 with these 4 levels of metabolite identification. Level 1, 2, 3, and 4, with level 4 essentially corresponding to unknown features, things that you can't really identify. Level 3 corresponding to compounds that are putatively identified by formula matching or high-resolution M over Z matching. Level 2 also corresponds to putatively identified compounds, but these correspond to MS-MS matches or EIMS matches. That is a spectral match to some database, and perhaps also using information about retention time or retention indices to confirm that. Level 1 is something that's almost never achieved unless you are doing targeted metabolomics, where this corresponds to positively identified compounds where you actually have an authentic standard that you've spiked into your sample or have as part of your assay to ensure that whatever you've putatively identified is exactly that compound. So level 1 is typically reserved for targeted metabolomics. Level 2 and level 3 are typically reserved or typical of untargeted metabolomics. And when people are doing level 3 compound identification, often they use something like formula matching or mass to charge, M over Z, M over Z matching. That's level 3. The favorite reference databases that people use are typically the ones with the largest number of compounds. ChemSpider with 68 million chemicals and PubChem with some 96.5 million chemicals. Now again, these are simple tools, concepts where you're trying to simply match a high resolution mass to charge ratio with some known mass or known chemical formula that you can extract from a high resolution mass spec, mass to charge analysis. A better method, obviously higher up the scale, the level 2 identification, involves spectral matching. And some of the favorite spectral databases that people like to use are resources such as Metlin or MONA, the Mass Bank of North America. Metlin is a large collection of compounds. Many of them are tripeptides and spectra that were collected on multiple instruments. The database has gone through a significant expansion with a number of spectra that have been predicted based on the compounds that have been entered. MONA, similar sort of thing, some 70,000 compounds, 200,000 plus spectra. Many of those are also predicted spectra. Perhaps a quarter of them are actual real spectra. If you look through other resources, there are other kinds of databases that also have large collections of tandem MS spectra. Human Metabolome database with about 300,000 MS spectra for 114,000 compounds. MZ Cloud, close to 200,000 spectra. The NIST database, more than 600,000 spectra, and on down the list. What's illustrated here are also the number of compounds, and you can quite clearly see that in many cases there are anywhere from 3 to 30 spectra per compound. These might correspond to spectra on different instruments, different platforms, different collision energies. But that's essentially the idea, to give you an indication of the number of spectra and also the number of compounds. Now, in the case of HMDB, the large number of compounds, again, many of those spectra are predicted. Overall, there's a relatively small number of compounds where actual experimental spectra have been collected. So whether it's things like PubChem or ChemSpider and doing formula matching, or doing spectral matching with MONA, Metlin, or HMDB, how well do we do? Well, we actually do pretty badly. When it comes to compound identification at level 2, when people are analyzing human biofluids with untargeted LCMS, typically the better labs are able to get between 150 and 250 compounds that reach level 2 out of perhaps 10,000 features they see. Using formula matching, Typically, they can get between 700 and 1,000 tentatively identified compounds, again, out of 10,000 features. So that corresponds to a very small fraction of the actual number of features seen. A more detailed study was done in 2015 by Emma Szymanski, where they looked at not biofluids, but actually river water, which contains actually a lot of human biofluids that have been processed. And the number of compounds that they could identify, and this is a multi-center trial, 649 compounds that reached level 1 or level 2 identification out of 8,500 features. So again, we're typically looking on the order of between 7 and 8% of features being identifiable in untargeted LCMS, and that number may in fact be somewhat less if we have high standards of, say, level 1 or level 2. So why do we do so badly? Especially with general metabolomics, human metabolomics, exposomic studies. Well, for one, there's an enormous chemical space out there. We know just from simple chemical formula comparisons that there could be potentially up to 32 billion compounds that would be potentially metabolites. Of those, if we use information about valency and sort of a natural state of things, perhaps 2.5 billion compounds are chemically feasible. Of course, in PubChem, there's almost 100 million compounds. If we look at all of the spectral resources from GNPS and MONA and METLIN, there's about 1.5 million spectra. So it's only a tiny fraction of the number of compounds in PubChem, about 1 or 2%. And these spectra have been collected on many, many different platforms. Now, if we look at the information a little more closely, we find that even though there's lots of spectra, there's only about 80,000 chemicals where spectra have been collected, and most of those are actually collected for EIMS. A lot of them correspond to derivatives, TMS derivatives. A lot of them correspond to special pollutants and environmentally interesting compounds. If we look at the situation for the high-resolution LCMS spectra, the number of compounds actually is much less than 80,000. It's probably on the order of 20,000. And of those, perhaps 1,500, maybe 2,000 are actually biologically relevant. So we've gone from 32 billion potential structures to a situation where we maybe only have useful spectra for useful compounds corresponding to about 1,500. So if that's all you've got for matching, no wonder we're only able to get 600 or 700 compounds in any given untargeted analysis. So why are we doing so badly? Well, one, obviously the chemical space is large. We're not able to search it. But it's also a situation that many people believe that, we just simply need to get a larger version of ChemSpider or PubChem. And that's actually a false hope. Many people are not aware that probably less than 0.2%, probably even less than 0.1% of the compounds in PubChem or ChemSpider have ever left the lab. They've been synthesized at milligram, maybe gram levels. A paper was published and then the compound largely disappeared. It never left the lab, so it can't be in human beings, it can't be in the environment, it can't be in plants or microbes. So these compounds, 99.9% of them, are never going to be found in a metabolomic study. As I mentioned before, there's only about 1,500 biologically relevant, environmentally relevant compounds that are available from most vendors. These are the things that you would find in biological systems or that have been released into the environment. And this simply reflects the fact that synthetic chemistry is hard. We just don't have large collections of compounds for purchase or for reference collection. What's also evident is that people are largely using the same collection of compounds and just simply collecting spectra on their favorite platform. And so the proportion of spectra keeps on climbing, but the number of compounds has stayed relatively static. So it's kind of useless information that we keep on adding to our databases. But it's also evident that regardless of how large or how small PubChem, ChemSpider, MONA, METLIN ever get, is that we still don't get very significant matches. Most of the unknowns are not in PubChem, they're not in ChemSpider, they're not anywhere. So we're missing an enormous number of compounds, even though we can identify clearly their mass, even though we can identify their MS-MS spectra, but we're not being able to identify them. So the question is, what are these unknowns? And the speculation is growing that in fact the unknowns represent biotransformation products, compounds that are metabolites of metabolites. We actually have a pretty good understanding of the types of compounds that are in our bodies, as we know from biochemistry, the types of things that we're exposed to, drugs or cosmetic chemicals or food components, those are mostly known. But as soon as they hit our liver, where phase 1 or phase 2 biotransformations happen, as soon as they hit our gut, where the microbial or microbiome transformations occur, things happen to these compounds. They are changed. And these produce, if we want, metabolites of metabolites. They can represent transformation products of the endogenous metabolome, transformation products of the food metabolome, transformation products of the drug metabolome, of the pollutant metabolome. And it's largely argued and believed that those represent the bulk of the unknown compounds. So what can we do as a community trying to solve or identify these unknowns? Well, one possibility is that we can systematically identify or predict what these compounds might be and actually synthesize them. We could isolate all the compounds, the drugs, pollutants, food components. We could not only synthesize them, we could also run them through artificial guts or artificial liver, microsomes, transform them, isolate those compounds. And we could do this for hundreds of thousands, some current estimates of up to 5 million compounds. Now, if we were to do this really cheap, with lots of graduate students working as slaves for long periods of time, we might be able to do it for perhaps $1,000 a compound. So if we take 5 million, multiply it by 1,000, we're looking at a cost of about $5 billion to essentially characterize the entire human metabolome. That will never be funded. It'll never be supported. It's probably also not even feasible. So the alternative that we're left with is to try and do the same exercise computationally. And in fact, that is probably more reasonable. And this concept of computational generation of metabolites or computational generation of observables like mass spectra is called in silico metabolomics. So it's an emerging concept to identify the unknown unknowns. It's computationally based, and it's based on the fact that we will never be able to isolate or synthesize most of the compounds that we are interested in. The concept is based on in silico or computer-based prediction of biologically feasible metabolites. It's trying to model the transformations that occur in the liver or the gut or in the environment. And it's also based on the in silico prediction of corresponding observables of those compounds. So the retention indices, the retention time, the mass spectra, molecular weight, collisional cross-section, or infrared spectroscopy. So this is illustrated with this diagram here, which we start in the upper left corner where we can take what we know. These might be compounds that are existing in various databases, things like the Human Metabolome Database and others. We take those structures and we run them through a computer program or programs to predict their metabolic transformation products. These create our unknown unknowns. And from this collection of structures, which might go from perhaps 250,000 of which we know to perhaps 5 million, we then calculate the observables. So we might calculate their NMR spectra, their tandem mass spectra, their EIMS spectra, their collisional cross-section, all the things that we can measure. So in doing this, we're able to create a large virtual in silico database of not only compound structures, but also reference spectra. When we have that database, we can run it through our standard protocol, which is, okay, you have your biofluid of interest or your river water of interest, you run it through your untargeted protocol, and instead of simply looking up these relatively small databases, you'd now be able to look at a much, much larger database containing biologically feasible structures and relatively accurately predicted spectra. And then you can do your compound identification. So how do we do in silico metabolomics? Well, the first thing we have to do is catalog what is known. So to know about the unknowns, we have to know about the knowns. So for the last 10, 12 years, my group at the University of Alberta has been building out a large collection of metabolomic databases covering a wide range of categories, organisms, purposes, applications. And I'll cover some of these very briefly. Perhaps the one that most people are familiar with is the Human Metabolome Database, or HMDB, something that we've been developing since about 2007. The latest release, which came out in January of this year, now has about 114,000 compounds, including quantified compounds, detected compounds, expected metabolites. It also has information beyond just the metabolites. It has pathways, it has MS, MS, and GCMS spectra, NMR spectra, referential spectra for the pure compounds, both experimentally predicted and computationally predicted. It also has lots of other data about concentrations, metabolite gene or SNP associations, metabolite drug associations for pharmacometabolomics. And then the fact that metabolites often vary with age and gender. That information has now been gathered. At some point in the next month or so, we hope to add another 75,000 compounds to HMDB, so that'll bring the total close to 200,000. We have another database which is actually more widely used than HMDB. It's called Drug Bank. A new version of this one just came out again in January of this year. And this is intended to cover, if you want, the drugome, the fact that people take both legal and illegal drugs, and that in any given population you'll often find several hundred different types of drugs in things like urine or blood. So this database catalogs the known drugs. It also catalogs information about experimental drugs. These would not likely be found in humans, but also has a lot of information about drug metabolites. And there's almost 1,400 drug metabolites. There's also referential spectra for those compounds. It also has lots of information about drug targets, and that's probably the reason why the database is particularly popular. People not only take drugs, they also eat food. And so a major effort over the last few years from our group is to try and catalog the chemicals that we find in food. Now these include things like food additives, chemicals that are deliberately added, but also the food constituents, the phytochemicals that are found in salads or fruits and vegetables, or in beer or wine. Right now, the database contains almost 30,000 different compounds, including lots of concentration values, lots and lots of reference information, lots of relationships between compounds and their health effects and their flavor attributes. But just like HMDB and Drug Bank, we've also included lots of referential spectra. Another database that we've maintained is the Toxic Exposome database. This is to help with exposure studies, and it's to collect information about things like pesticides and herbicides and other additives, solvents, carcinogens, drugs that are known to be toxic. Information that we've gathered from the ToxCast studies by the EPA. So this is covering generally compounds that are very low abundance, but certainly are known to be and have been found in humans and other animals. So right now it tracks about 3,600 compounds, has a lot of reference spectra, also has lots of information about their targets. So these are examples of databases covering the compounds that we expect or know should be in human beings or animals or fish or amphibians or in our environment. We've also been expanding T3DB to include another resource. It's called the Contaminant Database. So these are compounds that are not necessarily toxic, but they're found everywhere. And these include compounds you'll find in cosmetics and shampoos, stuff you'll find on clothing, fire retardants, surfactants, stuff that usually ends up in our drinking water, stuff that ends up in our dishwashers and clothing washers. The database is growing. Right now we're close to 80,000 compounds. We have lots of predicted spectra for this. It's under construction, and one of the surprising things is that most of the compounds in Contaminant DB are not in any database. People simply do not enter this information. Some of it's kept as guarded secrets by the cosmetic industry or the industrial chemical enterprises. But it was quite surprising to us to see how few of these compounds are in anything like PubChem or ChemSpider. We've also been expanding our resources to include plant compounds, plant-derived compounds, and this grew partly from our work with the food database when we discovered lots of compounds that are plant-derived, but there are also many other compounds that we can find. Some of them might correspond to medicinal plants, some of them might correspond to more exotic foods, not traditionally used in the Western world. But they do represent a large class of compounds, and we've been trying to expand this set as well. Again, many of the compounds that are in this database, which is under construction, are not found anywhere else. So it certainly tells us that there's plenty of compounds that are not in the referential databases that are biologically relevant, that are ecologically relevant, and that we as a community clearly can do better about making sure that those compounds are available. So this represents the compounds that we know, something on the order of 250, maybe 300,000 compounds. So the question is, what can we predict? And so this goes back to this question or this diagram that I was illustrating where we had, say, 250,000 known compounds. Now we have to predict how those compounds are going to be metabolized in our liver, in our intestine, in the environment, or just through processes that we call promiscuous enzymology. And this is where I'll dive into a little bit more about the details of how you do this. Now, if you're not familiar with biotransformation, this is sort of an illustration of how a drug can be transformed depending on where it goes. It can go into the liver, and it can go through phase 1 or phase 2 transformation, where it changes the structure of the molecule. Enzymes, the cytochrome P450s, or other phase 2 enzymes, glucuronidases and others, will modify the structure of a xenobiotic. So you can see some of the modifications that the cytochrome P450s will do in the liver, or the modifications that the gut microbiome will do in the intestine. They are very good at opening rings. Now, the transformation of chemicals, particularly drugs, has been studied for many, many years. And in fact, there are a number of commercial software tools that are around to do exactly that. Things like Metabolizer, MetaboExpert, ADMET Predictor, and probably the best known tool is called Meteor Nexus. All of these tools were developed in the 70s and 80s using sort of pseudo-rule-based, pseudo-artificial intelligence techniques to help the drug industry. The tools are expensive, and they were designed specifically for drug biotransformation, not for general metabolite transformation. And there are also lots of issues with these tools, not only because they're expensive, but they have a very limited scope. They weren't designed to predict microbial transformations. They don't usually predict phase 2 transformations. And they have no limits. And so they could create a propagation tree that can quickly produce literally millions of mostly unfeasible structures. As a result, we decided to start working on our own process and program to do something that was free. It's called Biotransformer. And it's to do a little bit more than what these other commercial tools do. It's to calculate or predict the biotransformations, not just for phase 1 or cytochrome P450, but also for phase 2, for microbial or gut transformations, for promiscuous enzyme transformations, and for environmental transformations that can occur when something is simply sitting in water or in soil or on your kitchen table. What it does is it uses a large database of transformations that we've collected, heuristic rules that we've built, and also some machine learning. And we've done lots of testing, and in fact we found that it performs much better than the commercial tools, and I'll explain that a little later. The tool itself is available, and a manuscript is under review right now, describing Biotransformer. The tool itself has, I said, these 5 components where it does phase 1 transformations, 160 some transformations are modeled in the software. Environmental transformations, about 300. Human gut or microbiome transformations, a little over 200. Phase 2 transformations, about 70. And then promiscuous enzymes conversions that'll happen, about 400. So we'll dive in a little bit more about Biotransformer, and this just tabulates the types of transformations and the sorts of transformation rules, about 1,200 that have been tabulated, and then a variety of reaction preference rules which help guide the process so it doesn't go to some combinatorial explosion. This is just an example of where we're taking a compound, quercetin, which is found in things like onions and apples, and running it through biotransformer. And you can see on the order of about 10 different compounds that are predicted through this biotransformation process. And in fact, many of these compounds, in fact, were found or are known. And you can go all the way from quercetin to hippuric acid, which is the typical end product for most polyphenols. And it is able to predict that. It's also able to predict not only the structures, the transformation products, but the enzyme or enzymatic processes. So you can actually draw a schema illustrating what's actually happened. Now, what we wanted to do was assess biotransformer because it's fine and dandy if you can generate structures, but are these actually accurate? And so we ran a bunch of different tests for biotransformer, comparing it to other tools, for instance, Meteor Nexus, which is considered the gold standard. And this is just simply illustrating what we took for 40 compounds, drugs and pesticides for which Meteor Nexus had been specially trained, and applied those same things to biotransformer. This is just illustrating what we call the precision and recall, or sensitivity and specificity. Perfect precision recall is 1. And what we can see is that neither biotransformer and Meteor are perfect, but biotransformer generally outperforms Meteor substantially, both in the level of precision and in recall. We've also taken biotransformer and ran it through other compounds and compared it to other tools. One here was ADMET predictor, just looking at human cytochrome P450 metabolism, and again the recall is in the high 90s and the precision is around 45–50%. So again, in this evaluation, biotransformer did better than the commercial tool. And then we ran biotransformer looking at endogenous metabolites. Now these are something that other tools fail miserably at because they weren't designed to train or trained with these ones. But based on the data that we've collected and assessing the performance, at least with endogenous metabolites, both human and gut microbial transformation, biotransformer is actually even more accurate. Recall is almost 90%, precision is almost 70%. So as I mentioned, this is a free software tool. It's available, and the website is up there now, actually. And as I say, the paper is under review. We've added tools for compound identification, where people can input both their mass to charge ratio and the presumptive starting molecule, and it will identify potential matches. What we've done in a variety of tests, we found that any given single step reaction will generate about 5 times as many compounds, and if we run biotransformer through 2 steps, we'll get about 20 times the number of compounds. So based on these calculations, we're predicting that the number of compounds that could be generated through biotransformer will be upwards of 5 million biologically feasible compounds. Biotransformer is relatively quick. It takes about 5 seconds per compound, and we can run it in parallel. So in fact, sometime over the next few months, we'll be generating about 5 million biologically feasible compounds through this particular software tool. If you have questions, I've given the developer, Yannick Jumbu Funeng's email address, and he's happy to help people with using this particular software. So going back to that diagram, we're showing how in silico metabolomics is done, where we start with the knowns, we transform them with biotransformer to create the dark matter or the unknowns. But then from those structures, we want to then try and see if we can generate observables. And this is where we use tools to predict the observables, namely the mass spectra. And my group has spent the last few years developing a number of tools to accurately predict tandem mass spectra, as well as EI or GCMS spectra. And the tool that we've developed is called Competitive Fragment Modeling, or CFMID. Now this tool uses machine learning. It's got a training set of high-resolution MS spectra where the collision energies have been ranging from low to medium to high. It naively fragments compounds, and then it learns from its mistakes how to properly fragment compounds and then generate the appropriate MS spectra. It uses a tool called a hidden Markov model or a probabilistic graphical model, and this is very good at recognizing patterns. And with any machine learning tool, the more training data you get, the better the performance. So these are just some plots comparing the performance of CFMID to other tools such as MetFrag or FingerID, and looking at different types of compounds, things that like di and tripeptides. You can see that it gets, the blue line indicates that it's very accurate, and that it's able to rank the top spectra as either the top about 60% of the time or the top two about 80% of the time. It can analyze compounds from the Metlin database or compounds from Mass Bank, and again, typically it's able to rank the top hit at around 80% accuracy. If you have additional information, particularly if you're able to collect your tandem mass spectra at not just one fragmentation energy, but at multiple fragmentations, you get better. Likewise, if the database is smaller, the performance for compound prediction is generally better. So if you have a smallish database, CFMID is able to correctly predict the compound and match the spectra about 80%. As you grow the database size, performance tends to drop, which is what would be expected. Now, CFMID is available as a server, and it does several things. It can take a compound and predict the tandem MS spectra. It can also take experimentally collected MS spectra and annotate those peaks, identifying the individual fragments. And it can also take tandem MS spectra and identify a compound. It does that by matching its spectra against those from HMDB and from KEGG. CFMID has been evaluated, tested, thrown into competitions for a number of years, and it's done very well in terms of these competitions, and it's been integrated in other software tools for spectral prediction. This is just an example of what would happen if you were, say, to take a spectrum and annotate it with CFMID. This is for serine, the amino acid, and it's just showing what the fragments are and how it calculated what they would be, and it illustrates them. You could do the reverse. You could say, here's serine, what's the MS spectra supposed to look like, and it would generate largely the same image. We've been upgrading CFMID over the last year. In particular, a new version, version 3, which will be released in December, has been designed specifically for predicting lipid spectra. And it also has better tools for matching and ranking both spectral matches and structure matches. We're also reworking CFMID quite substantially from the back end, making it the database, the training set much larger. We're including Orbitrap spectra. We're improving what's called the generative function, the fragmenter, and the performance already looks to be about 30 to 40% more accurate than the previous one. I'll just show you some examples, or at least an example of how the lipid version of CFMID, this recent version, is able to predict lipid spectra. On the right is what the version 3 is able to do in terms of its prediction. So the red spectrum is the predicted, the blue one is the actual one. We can rank or evaluate the score, what's called a Jacquard score, which matches peaks to peaks, and this Jacquard score is 98% correct. On the left is a tandem spectrum predicted using Lipid Blast, same compound, same lipid. In this particular case, Lipid Blast didn't do very well. It had a Jacquard score of about 28%. Normally, Lipid Blast is much better, but this is an example where sometimes the spectral predictions don't work particularly well. Nevertheless, lipid HMDB, the CFMID version, is able to predict very accurately for the 10, 20, and 40 electron volt collision energies, something like 28 different lipid spectra, 28 classes of lipid. And so we've applied this to most of the lipids in HMDB. So this is an example where we're taking tools, developing tools to predict observables. Now, tandem mass spectra are not the only thing we can collect. In fact, we can't really fully identify compounds only by their tandem MS spectra. We need to be able to include other things like retention time or retention indices or collisional cross-section, or there are now tools to collect infrared or infrared ion spectra. These, when brought together, or also including NMR, would help with formal identification of these unknowns. And in fact, there's efforts going on in several labs around North America and in Europe to come up with tools to predict these things and to do so quite accurately. So where are we? So we have methods or collections of databases, perhaps 250,000 compounds that we know should be in, say, the human body or should be in the environment. There are tools such as biotransformer, which are able to take those known compounds and to generate the biologically feasible structures. And then there are tools like CFMID or NMR prediction tools or collisional cross-section tools that can predict from a given structure an observable. And that's the observable that we ultimately experimentally measure. And so by combining those things, biotransformer, CFMID, and others, we can get back to this point of identifying the unknowns through spectral matching or database matching. Now, this may all seem nice as a theoretical construct, but in fact, there are a couple of labs that are already doing this. They've actually implemented this sort of pipeline into their processes. Groups in Switzerland, as well as Lloyd Sumner's group in Missouri, are actually using these tools or variations of these tools to identify unknowns and to create pipelines for doing this in a relatively rapid basis. Perhaps one unknown being identified on an average every day or every 2 days, where again the concept of in silico metabolomics, generating feasible structures, generating measurable observables, and then comparing the observables to the experimentally collected data to determine what these compounds are. So as I say, these are really happening. People are implementing in silico metabolomics, and these tools that I've outlined and variations that some of these groups have developed within their own labs are permitting this. So to wrap up, I think I wanted to emphasize that compound identification is hard, especially with unknowns, and doing it only by MS is even harder. I think I've tried to highlight that we do not have sufficiently large tandem MS spectral libraries or compound resources to do unknown identification, and that unless we have a change in thinking, we will never have sufficient resources to do absolute experimental identification. However, if we make the shift to using in silico methods, it offers a route to being able to identify, maybe not with the precision or accuracy we might all want, but it offers a possibility of identifying unknowns and getting around the problem of inadequate spectral libraries or inadequate collections of compounds. Now, the predictions of MS spectra, predictions of NMR, retention time, or collisional cross-section are still imperfect, but they are getting progressively better every year. And I predict, certainly within the next 5 years, that the accuracy of these tools will be such that they match almost what we could collect if we had the actual compound and the actual instrument. And if you don't believe that this process is working, I've tried to give you some examples of at least 2 groups, and I think the number is growing, using these in silico methods to perform relatively rapid and relatively robust unknown identification. And they're doing this so particularly in natural product analysis. So to wrap up, I just want to thank the contributors to this project, people who contributed not only to the databases, but also to the tools such as biotransformer and CFMID, people who've been working very hard in my group to annotate and to fill out the resources that hopefully some of you are using. And also thanks to the funders who have supported this project or these projects over the last 7 or 8 years. Thank you very much.